welcome at a very special seminar, which we consider to be like our Thursday seminars, but not in Thursday, but on Monday. And the reason we organized the seminar is that Dan Rabosky is here. So it's my great pleasure to introduce Dan Rabosky. Dan Rabosky is top expert on diversification, evolutionary diversification, macroevolution, and related stuff. Actually, his works hugely inspired the stuff we did with Anthony Machach here in last several years, because Dan started to think and explore the things like diversification slowdowns and diversity dependence and so on. And today he will be speaking basically in the same line. He will be speaking about diversification and the latitudinal diversity gradient. Please.
Thanks very much for that intro. Thanks for coming. So I thought I'd give a talk today that just had to do with some things that relate to one of my main interests, which, even though we've done a lot of stuff to look at diversification rates, estimating speciation and extinction, ultimately what I'm really interested in is explaining patterns of species diversity at different time scales. So today what I'm going to focus on is the latitudinal diversity gradient, and a little bit about what we can learn in terms of speciation and extinction rates and what role they might play in the latitudinal diversity gradient. And really, the latitudinal diversity gradient in some ways is something of a tropical diversity puzzle. And I think a useful starting point for thinking about the latitudinal diversity gradient is thinking about what the Earth looks like from space. So if you were like an extraterrestrial visitor to Earth and you came and saw this view of Earth, you would see a couple of things from inspecting Earth at this sort of spatial scale. One of which you could infer is, because there's blue and there's white and there's land, you would infer some things about chemical processes, maybe relating to microbial and feedbacks and photosynthesis that basically gave us this blue and white marble that we see. Second thing you would notice that has to do with life is that some areas are brown and some areas are green, and you can see that from space. So we know basically why that happens. There are areas that have productivity and areas that don't. And so, but what you wouldn't see if you were looking at Earth from space is another view of Earth, which is basically has to do with how species richness is distributed across the surface of the Earth. And that is not visible in this sort of framework. But if you could imagine replacing that Earth with one where we've actually made it sort of a heat map of species diversity across the surface of the Earth. So what I'm showing you here is just an illustrative example for one group. These are marine ray fin fishes. And what you can see is that there's basically warm colors like red are basically areas where species diversity is high, and blue colors are basically areas where species diversity is relatively low. And so species richness, species diversity, I'll use them kind of interchangeably through my talk, ranges from places like in the tropical Indo-Pacific where you might have, an 100 by 100 kilometer cell, you might have something like 3,500 or 4,000 species. And then you go to places like the high Arctic or the Antarctic where you might have 20 or 15 species at the same scale. So this kind of general pattern, meaning namely the idea that there's variation in species diversity, applies to basically every group of plant, animal, microbe that you could think of. And more often than not, the patterns are consistent with what you see here, where places that are warm, like in the tropics, have lots of species, and places that aren't do not have many species. Now, this is another illustration of the latitudinal diversity gradient, this latitudinal diversity gradient, or this variation in species richness as we go from high latitudes, like the temperate or boreal zone, versus tropical regions. So these are basically the numbers of tree species on a log 10 scale for 11 major biomes. And so we have 3 tropical biomes. So these are the Afrotropics, the Asian tropics, and the Neotropics. And you can see that they stand way out in terms of, and we're basically correcting for the area of those biomes here. And what you can see is that the tropical areas stand way out relative to the temperate and boreal biomes, which have relatively few species of trees. Now this figure was published by Paul Fine and some collaborators in 2008. What I really like about this figure is that it's plotted on a log 10 scale, because actually if you unlog the axis, it really brings the pattern out. So if you think about tree diversity on an unlogged scale, you can actually see that basically, to a first order approximation, outside of the tropics, tree diversity is basically statistically indistinguishable from zero. So really, you're really talking about a planet that is basically dominated by diversity of tropical trees in terms of actual numbers of tree species. And then you got this like small little crust of species everywhere else, which is basically what we get left with in in central North America and in Europe. So if you go to an oak hickory maple forest close to where I live, so this is a reserve about 20 kilometers from my hometown of Ann Arbor, Michigan. And if you were to take about a 50 hectare area in there and ask how many species of trees do you have, that was about 41 tree species per square kilometer at that sort of spatial scale. Now conversely, if you were to go to a patch of lowland tropical rainforest like Yasuni National Park in Ecuador, you might find something like almost 1,100 or more species at the same sort of spatial scale. So this is in 1 square kilometer, you can find more than 1,000 species. So there's about a 20-fold or more increase in species diversity relative to what you would have in Michigan. So it's a huge difference in species diversity. And so what we're going to talk about today is really what causes this large-scale variation in species diversity. So this is one of the fundamental biodiversity pattern on Earth, this contrast in species diversity between warm, wet areas in the tropics and cold regions outside of the tropics. So people have obviously been studying this for a long time. We're going to ask what we can learn from thinking about this in light of some fundamental mechanisms and how we might distinguish these mechanisms and in terms of how they might cause this gradient. So I think it's useful. There's a lot of ways that we can think about what causes the latitudinal diversity gradient. And I think it's useful to think about the pattern. So in fact, not only are there a lot of ways, there's a confusing array of potential mechanisms. So you'll often see people cite figures or say phrases in papers like, there are dozens or hundreds of causes of the latitudinal diversity gradient. And so how do we make sense of all these potential mechanisms for the latitudinal diversity gradient? Well, I think it's really useful to think about the fact that, get rid of this thing. That's not going to work. I think you can really distill most of the potential causes of the latitudinal diversity gradient into just 3 very general sets of mechanisms, within which the vast majority of other ideas are nested. So here's a schematic showing how diversity might originate and how these sort of generalized conceptual processes might contribute to a latitudinal diversity gradient. So let's imagine we're standing here in the present. So this is time on this axis. This is species diversity right here. You're an observer. This is like your observation frame of reference, the present day. So now let's imagine that the tropics are basically this orange diversity accumulation curve, and the temperate zone is this blue diversity accumulation curve. So the simplest idea for why there is a latitudinal diversity gradient is simply the idea that there has been more time for species diversity to build up in the tropics relative to the temperate zone. So all things being equal, even if the rate of diversity accumulation is the same for the temperate zone and the tropics, if the tropics have had longer for diversity to build up, they're going to get to a higher point by the present day. That right there will give you a latitudinal diversity gradient. This is probably the oldest hypothesis for the latitudinal diversity gradient. This goes back to the 1800s. Naturalists like Wallace were speculating about how the stability of the climatic stability of the tropics could lead to their exceptional diversity. But this is not the only possible model. You could also imagine that maybe the tropics and the temperate zone are the same age, but actually diversity accumulates faster in the tropics relative to the temperate zone. And so this is basically a hypothesis about the rates of species accumulation through time, or a diversification rate hypothesis. So basically the slope of this curve is steeper for the tropics than it is for the temperate zone. Obviously, these things can combine in different ways, but this is just sketching out sort of the simplest set of potential causal mechanisms for the latitudinal gradient. And the third hypothesis is one where you basically have sort of an equilibrium mechanism or a diversity-dependent mechanism where there's diversity-dependent feedbacks between speciation and extinction that cause species richness to be regulated at different levels in the tropical and in the tropics relative to the temperate zone. So this, one way of thinking about this is, one interpretation of this would be sort of a carrying capacity kind of argument. So this could be the idea that there's more niches in the tropics relative to the temperate zone. And there are other interpretations of that. So what we're really going to focus on today is these two hypotheses right here, which are basically non-equilibrium hypotheses. And the reason these two hypotheses are non-equilibrium is because these two hypotheses, unlike the diversity-dependent hypothesis, is that these hypotheses predict that if we were to sit here today and imagine that the processes that are in operation in the modern world continue running exactly as they are into the future, the latitudinal diversity gradient will continue to get even steeper. And it will get steeper because the tropics will continue to accumulate even more species, or because they'll accumulate species diversity even faster. Now, the equilibrium model right here basically does not really predict this, that the latitudinal gradient is going to change if environmental conditions stayed the same as they are today. So that's why there's an equilibrium versus non-equilibrium approach to thinking about this. Now, and specifically, we're going to focus on this non-equilibrium hypothesis in particular, this idea of diversification rate. So here we have differences in diversification rate, of course, is the outcome of the balance between speciation, which creates diversity, and extinction, which removes diversity. So you have the balance of these two processes. The difference between them, speciation minus extinction, is what we call the net diversification rate of an evolutionary process. Now, one thing when I said a few slides ago that there were hundreds of hypotheses for the latitudinal diversity gradient, one thing to keep in mind here is that when I say speciation rate, for example, speciation rate as a cause of the latitudinal gradient, like maybe speciation rate is faster in the tropics, that's not a single hypothesis. So there are many potential mechanisms that get subsumed or contained within this general umbrella mechanism of speciation rate. And so one idea is one that goes back a while in studies of the latitudinal gradient, is this idea that the chemistry of speciation, or evolutionary speed, is faster in the tropics. And so we go back to this 1992 paper, kind of a classic on this, and that we see things like the general acceleration of physiological processes with temperature, in addition to faster mutation rates and shorter generation times. These things must have significant effects on the speed of evolution, so evolution, or the speed of selection. So evolution will be faster in the tropics, and this greater effective evolutionary time in the tropics, because of that greater evolutionary speed, will lead to greater species numbers in the tropics. So that's basically the idea that processes just run faster, biochemical processes run faster, species interactions run faster, and all of that generates faster rates of speciation in the tropics relative to the non-tropics. But that's just one possible mechanism, and there are a number of others. So this is a widely cited paper from 2007 that sort of catalogs a number of evolutionary mechanisms for the latitudinal diversity gradient. And these ideas include everything from genetic drifts in small populations cause fast evolutionary rates. Maybe population sizes are smaller in the tropics. Maybe there's large areas in the tropics and it generates more allopatric speciation, so we have faster speciation through that. Maybe there's stronger biotic interactions in the tropics that lead to faster evolution of reproductive isolation via some sort of trade-off kind of mechanism, or correlated selection kind of mechanism. Now, we could go and parse any one of these explanations in terms of picking apart specific predictions. But actually, all of these mechanisms, all of these specific mechanisms, make very general predictions, and all of them predict that speciation rates should be fastest in regions of high species richness. And so this is a very general explanation for the gradient then that might have a number of potential lower level causes. But the first thing we should do if we're going to test, if we want to understand the latitudinal gradient, is ask, can we test this hypothesis, and are the data that we have consistent with the idea that speciation rates are faster in the tropics? So that's the starting point. If so, then we can look to the lower level mechanisms. If not, we don't need to look any further. And so what I'll ask today is whether speciation rates drive the latitudinal diversity gradient. And the first part of my talk, I'll focus on species richness and speciation rate in marine fishes. And then I'll talk about some patterns, some other taxa, and I'll close with just something of a rant on the future of biodiversity data. So a little bit of background for my talk, and that this is basically just something to keep in mind when you look at some slides with some figures with some phylogenetic trees and some speciation rates. So what I want you to think about speciation rates today, for the purposes of my talk, is I want you to think about them as a measurable trait at the tips of a phylogenetic tree. So we won't go into any details about how we come up with these estimates. You might imagine that you have a phylogenetic tree, like this phylogenetic tree of North American warblers. And we might use a particular statistical model to reconstruct evolutionary rates across that tree. So now I've colored branches with colors indicating that reconstructed rate of speciation across this tree. And what we're going to focus on analyzing today are what we're going to call tip rates. And this is basically just an estimate of speciation rates at the tips of the tree. And so this is basically saying, based on past history, what is our best estimate of the present day rate of species formation? And so that's basically the speciation rates we would have at this particular block here, because this is a time calibrated tree, and each of these tips is a species in this example. And so let's say, now I'm showing you these actual tip speciation rates. I apologize for this thing. I don't know why that's sitting there, but I can't clear it. But so we've got like a number of species. Like let's say we're looking at these species here, like the ovenbird and the Magnolia warbler. Well, this is the speciation rate for the Magnolia warbler. The tip rate, the best estimate of the present day rate, is somewhere around 0.3 lineages per million years. Our best estimate for the ovenbird is somewhat slower, somewhere around 0.2. That's just one example. And so we can obviously do this for any taxon in the data set. Now, how good are these estimated tip speciation rates? Well, as far as we can tell, that they seem to be pretty good, and they seem to be pretty robust to a lot of violations of their assumptions. And the results I'll actually show in my talk today, we'll be using a couple of different methods for reconstructing these rates. But this figure right here is just going to show you an example where we looked at how well these things performed across data sets simulated under a whole bunch of different kinds of scenarios. And basically going to show you the relationship between the true tip rates and the estimated tip rates across many tens of thousands of species from these really complicated phylogenies. And basically, just looking at a couple of different methods here. For example, a lot of the results we'll talk about today will focus on these rates from the BAM method. You can see there's a pretty strong correlation overall between the true rate of speciation and that which we estimate with the model. So anyway, speciation rates are generally well estimated at the tips of the tree. I mean, this is something that's been predicted for a long time, so this is not really a surprising result on some level. And the second thing is that when I say speciation, I really mean speciation. I don't mean net diversification. So net diversification is the difference between speciation and extinction, so speciation minus extinction. And I am actually talking about speciation rates here. Of course, that raises another question, which is, what about, one other point. So you'll also see some analyses like this right here. I'm going to ask whether particular kinds of traits like latitude are correlated with rates of speciation. And so you'll see some analyses where we look at a very messy cloud of speciation rates, and we want to see whether a trait like latitude actually affects the rate of speciation. Now, all that stuff is we treat with very specialized statistical approaches that control for things like phylogenetic non-independence between taxa and so on. So, and my lab has worked a bit on methods for doing these kinds of things. There's lots of approaches we can use, but just all you need to know is that there are approaches to deal with the non-independence of taxa, and that's something that's taken into account if I show you significant results in this talk. Now, the fact that we're talking about speciation immediately, and not net diversification, immediately raises the question of what about extinction? So for phylogenies that include no extinct species, it turns out that estimates of extinction for phylogenies that don't have any extinct species are actually pretty bad, even when the assumptions of the model are met. And extinction is very difficult to estimate without fossils. So we could happy to talk more about that later, but this is there are, in our field and macroevolution in general, I think you can sort of divide in some ways the spectrum of people into a couple of categories. And on one hand, I think you have some folks who believe that maybe we will just get better models and be able to pull information about extinction out of phylogenies that contain no extinct species, and other folks who think that that's unlikely. I tend to be in the more skeptical camp there, but in any event, I think we all agree that those kinds of estimates will get better when we have things like fossil information. This is just a simple example showing you just sort of the best case scenario, looking at your anyway, this isn't particularly important, other than to say that, like, if we're looking at clades of a given size and we look at how accurate our speciation and extinction rate estimates are in terms of the correlation with the true rate, this is under perfect conditions. This basically shows that our speciation correlation with the true rates is always better than the extinction correlation with the true rates. So, and in fact, the extinction rates get much worse when the model is actually assumptions of the model are violated. All right, so for the first empirical thing we're going to talk about here is the relationship between speciation rate and species richness in marine fishes. And so we want to ask is, does speciation rate variation generate spatial variation in species richness? And so this is a very simple idea. And so really what we're going to do is two things. First of all, we're going to characterize the geography of marine species richness, and then we're going to estimate speciation rates across extant fishes and ask whether the speciation rates predict patterns of species diversity overall. And this is a collaborative project that involves contributions from a number of other folks who were involved with aspects of everything from phylogeny reconstruction to spatial data compilation and so on. So this is a phylogeny that we generated for marine fishes. This has about, this is the overall phylogeny includes about 12,000 species with genetic data, and then we added the other 18,000 species of living fishes into that tree using a special kind of method for placing unresolved taxa on the tree using taxonomic constraints. It's a conservative way of putting them on the tree that lets us account for some biases in how we estimate rates of speciation from the data set. So this is just a reconstruction of speciation rates across this tree. And the only take-home message here is that there's places on this big tree that have fast rates, and there's places on this big tree that have relatively slow rates. So there's a lot of rate variation here. Now, why this is nice is because it actually means that if we're testing a hypothesis like speciation and latitude, we have lots of statistical power to work with here. There's a lot of rate variation. And this is just basically a rank order plot of speciation rates across all the species of fishes in the tree. And so what you can see here is that a lot of species have relatively slow rates of speciation, and then there's a small number of species that have fast rates of speciation. And so at one end of the spectrum, we have things like paddlefishes, which are sort of sometimes considered a living fossil. There's only a couple of, well, actually, there's only one living species anymore since the other one species went extinct recently. But they're at one end of the spectrum where they have very slow rates of speciation. Then you have things like these rockfishes from the Pacific Ocean, which have very fast rates of speciation. And sort of other famous clades like cichlids, for example, would sit over here if you're wondering. So in fact, a substantial chunk of this fast rate part of the curve would actually be cichlids from East Africa. So this is the overall picture of the geography of species richness in marine fishes. And so we had to it was a fair amount of work to actually compile the geographic ranges for this. There was no standardized database of marine fish spatial data. And so again, you have really high species richness in the tropical Indo-Pacific, relatively low richness outside of that zone. And this, if you look at species richness as a function of latitude based on midpoints of individual cells, you can see this is the equator right here, and this is getting south, this is getting north. See this nice relationship overall, sort of what we expect. And this sort of general idea has been shown. This general result has been shown previously using other kinds of data. And there's this one interesting area right here where really a huge amount of the marine fish diversity is actually concentrated, sometimes called the coral triangle. And so this is basically a region that is home to centers of diversity for a large number of tropical reef-associated clades. And a lot of people have been interested in this region. In fact, they've given it the name Coral Triangle to basically flag it as a big biodiversity hotspot. And there's a number of papers that have looked at this question of whether there's something special about this Coral Triangle region that drives speciation in taxa. So there are many, many papers that either approaching it from a phylogeography perspective or from a macroevolutionary perspective that ask whether there's something driving speciation in reef fishes in the Coral Triangle. Well, what I'm going to show you now is taking those speciation rates that I showed you on the previous slide. And what we're going to do now is we've got this mapping of species diversity in space. And now what I'm going to show you is a corresponding mapping of speciation rates in space given those reconstructed rates. And so if you haven't seen this result before, how many of you think it's going to be faster speciation in the tropics? Who thinks it's going to be faster speciation outside of the tropics? Who thinks the data are going to be ambiguous? No result. which is probably the most likely a priori outcome. So, yes. In fact, this is the relationship between speciation rate and latitude. So basically, across the board, in both hemispheres, both the northern hemisphere and the southern hemisphere, the fastest speciation rates by far are basically in regions that are basically close to the Antarctic or the Arctic. And it extends basically up and down in the sort of north temperate and south temperate zone. So completely opposite what a lot of people have been predicting. And completely counter the idea that speciation rate is the primary cause of high species richness in the coral triangle right down here. Note there's basically nothing going on from a speciation perspective right there. So we can look at this another way. So this is, actually we'll spend a few slides here looking at this different ways. But this is latitude right here, species richness. And this is basically reconstructed cell-based speciation rates using the rates reconstructed across the entire tree. And you get this nice sort of banana shape right here where basically you have this trough of speciation rate close to the equator.
If you say like per cell, so that's average? Or?
This is an average across cells. And I will look at it in other. This is basically the average rate across all the taxa that are in a particular cell.
Okay.
So yep. So these are averaged rates across all the species that are actually in the cells. And we've looked at this other ways. So we looked at like different quantiles of that distribution and so on. But there is a very different way we can look at it, which I'll come to in a couple of slides. So this is basically taking a cell-wide sort of average rate aggregate property. And so we can do a number of other things with this. We can say, well, because related to your question, what if because those rates are average rates, what if it's species with wide geographic distributions that are basically diluting down the rates for some regions? Well, that's an obvious concern. So one of the things that we did, we sliced the data into a number of different biogeographic provinces that basically restrict them to, and we restricted the analysis to core species that are only found in particular biogeographic provinces that are basically isolated from other provinces. And so we did this for the marine ecoregions of the world. So then we basically are looking at rates only for species that are found in the Antarctic region, for example, only for species that are found in the Caribbean and so on. So when we do that, we basically find the exact same pattern. So that controls for geographically widespread taxa actually driving the pattern. So we saw fast rates in the Antarctic, fast rates at north temperate latitudes. And so overall, is species richness correlated with speciation rate? Here's speciation rate on this scale. This is species richness here. I'll color cells based on the latitude that they are at. So tropical cells are basically red. High latitude cells are going to be blue. And so this is the overall relationship between species richness and speciation rate. So species poor cells over here with fast speciation, species rich cells over here from the tropics with slow speciation. And sort of immediately, I mean, it's nice to see this maybe, but I guess we could predict that we would see this. This is just the relationship between speciation rate and annual sea surface temperature, which sort of emerges immediately from this, but it's kind of nice to see it at this relationship right there. This is just taking the average annual sea surface temperature for individual cells and looking at speciation rate as a function of temperature. So that's one way of slicing. That's one way of slicing the data. We can take a completely different approach that does not do any averaging over geographic regions. And so this approach, we're going to actually look at rates for individual species. And so what we're going to do here is take this analysis right here, these results, and well, this is actually looking at rates for individual clades. So what I'm showing you here, of this whole tree right here with fast and slow clades, now I'm gonna drop away all of the clades that are not primarily centered on high latitude polar and temperate regions. So we're going to get rid of all the tropical species and just see what's left. And this is basically what we have. So remember, red is fast, blue is slow. And this is basically pulling out just a handful of clades that account for about 90% of all species diversity of fishes in the high latitudes. This is basically Arctic and Antarctic communities. And so you can see here you have rock fishes, fast rates. We have snail fishes, eel pouts, Antarctic nototheniioids, and basically a few other clades scattered throughout this tree. And basically all of these things, actually one of these this is a clade of flounders over here. But in every case, basically the high latitude clades that we pull out are characterized by fast rates of speciation. And you can just see it graphically here. There's not a single blue clade in here. Now, well, we can also do this more formally. So that's just a graphical sort of interpretation of the pattern. It's nice to see this. But we can actually take individual species and take the geographic midpoint of their range and plot that as a function of their speciation rate and do formal statistical tests on that. So basically, this is what you're going to see now is plots of all species with their individual tip rate against their latitude. And when we do that, we basically get a cloud of points that looks like this. Now, this is super noisy, and we know that there's a huge amount of estimation variance as sampling error just in these results right here. But we can fit models to this, and no matter what we do, we basically get the same result that we're getting at the cell stage. So this is basically the fitted relationship between latitude and speciation rate across all of the individual species in the data set. And no matter how we analyze it, we can analyze it using a couple of different methods that account for phylogenetic autocorrelation between species and their rate. And we get a highly significant relationship where the further you get from the tropics, the faster your speciation rate is if you're an individual species. And so put this another way, this is basically the median speciation rate across all species. We can break this down into a few individual clades, which I think is kind of informative on some levels, because we can look at like Antarctic ice fishes, for example. So this is basically a clade that dominates high latitude communities or Antarctic communities, so their perciform lineage. And you can see where those species basically fall out relative to the overall median rate. These are Sebastes rockfishes, a clade that's very dominant in high latitude northern communities. You can see they're basically a couple of southern species, but these things are well above the median in every case, almost every case. These are some deep water and high latitude lineage. These are eel pouts and snail fishes, also perciform lineages, and they also stand out as well above the median in general. The colors are basically oranges, eel pouts, and blue is the snail fishes, so these very bizarre sorts of morphologies like this. Now conversely, you can actually take the main iconic tropical reef clades, and this includes things like wrasses, puffer fishes, angelfishes, surgeonfishes, triggerfishes, butterfly fishes, gobies, all the sorts of things that would account for about 90% of the diversity in shallow water tropical reef systems in the tropical Indo-Pacific. And if you looked at those species, this is basically what you get. They rarely occur at high latitudes, and you get this giant mass of points that's either centered on the median or actually less than the median. So there's basically nothing going on in any of those traditional reef-associated clades. And so just to summarize this part of the talk, so overall, we have this negative relationship between species richness and speciation rate. So high latitudes, fast speciation, tropical latitudes with high species richness, slow speciation. And one of the nice things about looking at the pattern at this scale is that we can make some, well, granted this is just fishes, but I think fishes are good because there's we have enough power here and enough breadth of this data set to know that this is not just driven by one hemisphere. We see it in both hemispheres, which are actually dominated by different clades. It's not just driven by well-sampled clades, because there's well-known clades that have received a lot of attention and poorly known clades that are also contributing to both fast and slow rates overall. And we do a number of analyses of that as well, which I won't show you here. It's not driven by shallow water clades. The pattern applies to both deep and shallow water lineages at high latitudes. However, deep water lineages that are centered in the tropics do not have fast rates. If anything, they have slower rates than the average. And it's not driven by poorly known tropical faunas, and we use some approaches to try to correct for that as well. In fact, tropical fishes are probably better known. At least a lot of the sort of iconic reef clades, except for gobies, are probably better known than many of the high latitude clades, especially snail fishes. So let's talk about some patterns, some other taxa, because it turns out that people have been looking at this recently because now we have this sort of phylogenetic data to do things, and we also have the statistical machinery to ask questions and extract signal of speciation rate from some of those data sets. So this is a study that we did on birds a couple years ago. So this is the latitudinal diversity gradient in New World birds. So the neotropics are really where it's at for birds. The bird diversity in the neotropics is really unparalleled, and you have places in the neotropics where at a relatively small spatial scale you have 500 to 600 species. I do a lot of field work in southern Peru, in the Amazon, and there's about 500 to 600 birds that basically co-occur at the sort of 20 kilometer by 20 kilometer cell area in that region. But you contrast that to like the North American boreal zone or temperate North America, and species diversity is far lower. So these are breeding birds. So this is the relationship between speciation rate reconstructed across all New World birds and their latitude. And so what you're seeing here, so this is latitude on this axis. This is the equator. These are high southern latitudes. These are high northern latitudes. And this is basically just averaging the speciation rate and looking at the median speciation rate and a bunch of quantiles of the distribution for all the species of birds that breed at different latitudes. And so this, for example, right here is basically the 5%, 15%, and so on, median, all the way up to the 95% quantiles of the rate distribution for all species of birds that actually overlap with their breeding range overlaps with the equator. And so what you can see, there's really there's no trend in speciation rates overall. And so this is a result that's basically been confirmed by a number of other studies, and so this is not really something that had showed up in, the basic result showed up in 2012, and it's sort of been revisited a few other times since then, including by us. But now in the last couple of years, people have looked at this in a few other taxa as well. So just to give you a few examples here. So this is a paper by Economo et al. in last year looking at ants. And so this is the latitudinal diversity gradient in ants right here. And so you have this, again, this nice latitudinal diversity gradient where basically species richness centered on the equator. They reconstructed speciation rates across a phylogeny of ants, and they looked at how speciation rates stacked out with latitude. And they call it diversification, but really what they mean in this case is speciation. Or at least I'm going to interpret their diversification as speciation because it actually is statistically speciation. But they find no trend between speciation rate and latitude. So again, similar results to, actually different results from fishes, because in fishes we actually find a negative relationship between latitude and speciation rate, but they find no pattern. This is a paper that's on bioRxiv now, so I'm sure this will come out at some point in the near future. And this is basically an analysis of the relationship between latitude and speciation rates in plants. And so what you're seeing here are basically quantile or distributional quantiles of speciation rates for plants across the phylogeny of about 30,000 land plants. And they're factoring them by latitudinal quantile, and they're basically looking at the rate quantiles as well. So this is the speciation rate on this axis, and this is latitude here. So this is basically the tropical rate distribution, and these are the high latitude or temperate zone rate distributions. And so you can see that the tropical rate distributions are basically shifted lower than the temperate and high latitude rate distributions. So that's consistent with what we see in fish. This is another paper that's on bioRxiv now. So this is Nate Upham and Jake Esselstyn and Walter Getz, paper on bioRxiv this year. This is a really nice paper. They built this amazing phylogeny for mammals and then modeled diversification rates a whole bunch of ways. And they basically reconstruct speciation. They call it diversification, but it's actually speciation. And they basically look at the relationship between speciation and latitude. This is the latitudinal diversity gradient in mammals, this frequency distribution here. So they're starting now at 0 latitude and going, so now they're looking at the absolute latitude. So 20 right here could be 20 degrees north or south, right? So this is the equator, and this is getting away from the equator in either direction. So you have a nice nice peak in species diversity at the equator. Species richness drops off as you move away from the equator. However, there's either no pattern in speciation rate, or speciation rates actually show a weak increase as you actually move away from the equator. So again, these are all consistent with the basic idea that we've either consistent with the idea that there's no correlation between species richness and speciation rate in the living biota. So conclusions from this part of the talk. Okay, so we looked at some high latitude fish clades, and we basically find a trend towards fast speciation in high latitude grid cells and high latitude fish species themselves. And there's basically a weak relationship between speciation rate and species richness, and this should not read, at least for some vertebrates, because it's basically, at least for most taxa that have been looked at now. So this is just the relationship between speciation rates and frequency distributions of speciation rates for tropical and temperate passerine birds is one example. And so I think this is interesting to sort of think about what this is telling us about the latitudinal diversity gradient. Because really this is evidence against not just one specific hypothesis for the latitudinal diversity gradient. It's actually evidence against a whole large class of mechanisms that predict faster rates of speciation in places with the highest species richness. So I'll give you an example. So this is one specific idea about why speciation rates should be faster in the tropics. So there's a pretty well-developed body of theory now about speciation rates and temperature. Now, this is theory that's derived from first principles. It's not derived from data. And so the idea here, this is based on ideas from the metabolic theory of ecology. And the idea here is that we can basically think about a kinetic effect of temperature on processes that ultimately result in speciation that have to do with, well, there's a number of ways in which you might imagine it happening, but the basic idea is related to the evolutionary speed hypothesis that I mentioned early on in my talk. And so we can imagine that there's basically a temperature kinetic expectation for what speciation rates how speciation rates should vary as a function of temperature under the metabolic theory of ecology. So we can actually get a quantitative prediction of what speciation rates should be under the metabolic theory of ecology. Now, when you take the theory and you actually plug in some numbers, and you look at what speciation rate should be as a function of the temperature range that we see across latitude for marine fishes, it basically is something that looks like this. So we should basically see a relationship between annual sea surface temperature and speciation rate that's sort of on this scale right here. However, this is actually what we see. So what this basically tells us is that the observed data for fishes are completely inconsistent with the speciation rate predictions from the metabolic theory of ecology. This does not mean that the metabolic theory of ecology has nothing to do with the latitudinal diversity gradient, because there's a lot of other interesting things that come out about scaling and ecological interactions that might affect things like carrying capacities and so on that are in the metabolic theory of ecology. But it does tell us that this first order hypothesis about prediction about speciation rates is almost certainly wrong. So, and also we're not seeing evidence consistent with this from other texts as well. And so what do we actually know about the latitudinal diversity gradient? Again, we like to think sometimes, or we say that there are lots of hypotheses. How are we going to get anywhere with the latitudinal diversity gradient? But really, you could think about you can distill those down into just a core set of mechanisms. Time, rate, and diversity dependence. And here, I think we're basically converging on a point in our knowledge, understanding of this phenomenon, where we've basically managed to reject one of the major categories of explanations, which has many, many sub-hypotheses that people have been arguing about for a long time. And we're basically getting to a point where we've largely rejected this specific idea. Now, the idea that extinction might be lower in the tropics is another sort of issue, and I'm not going to get into that. But my view about things like the latitudinal diversity gradient is that rather than thinking about showing that something is the driver of a pattern, what we should really try to do is think about the ways in which we can make tests that strongly reject specific hypotheses and test them. And so that's what I think we can do with speciation rates. Even if we found faster speciation in the tropics, that doesn't mean it's the cause of the pattern. However, failing to find faster speciation in the tropics means we can reject that class of mechanisms. So the last couple of minutes, I'm going to talk about something that's related, but in some ways completely different. So this is the rant part of my talk. So maybe it's because I'm a museum curator at a major biodiversity institution. But one of the things I have to ask when we do these kinds of broad, synthetic kinds of comparisons is our knowledge of biodiversity adequate to actually answer questions like the ones we're actually studying here? And so I would say, well, maybe not. Taxonomic knowledge is insufficient for many taxa, maybe not so much for fishes, but if you get to things that don't actually have vertebrae, it gets really, really bad for some groups. Basic natural history data is lacking for many or most taxa across the tree of life. And long-term ecological data are extremely limited. So in some ways, you can imagine that our ability to even ask questions about the latitudinal diversity gradient is going to suffer because we don't actually have the data to deal with basic taxonomic and natural history kinds of issues that are important for parameterizing ideas about the latitudinal gradient. We like to think that we're living in this sort of modern era of biodiversity synthesis because we have these data aggregators out there like GBIF and VertNet and FishNet that have literally, in some cases, hundreds of millions of records of things. And so we can just go there, download some stuff, and what else do we need to actually know? Well, let's just take our fish data set as an example here. So across our fish data set, less than or only about 35% of the species in our data set are represented by fewer than 10 georeferenced museum records. About 75% have less than 100 records. And mind you, those are 100 records where 80 of them might have come from a single spot where people were actually doing a particular research trawl. And only 5% of species have actual quality information about trophic ecology associated with them on FishBase, which is sort of the largest aggregator of fish ecology and natural history information out there. So one of the things that we've been doing in my lab to sort of try to address this, at least on a local scale, to the extent that we can contribute to this, is to do some field-based studies of ecology of reptile and amphibian communities from a couple of really, really diverse communities. And our basic idea here is just trying to get some basic idea that lets us ask, basic data that lets us ask questions like why are there so many species of reptiles and amphibians in places like the Neotropics or Australia, which are the two places that we work. So this is a global species richness map for lizard and snake diversity. Consistent with the other stuff I've shown you so far, you can see there are places in the world that have lots of species and places in the world that have relatively few species. High latitudes again, relatively few species. Australia, not necessarily super high latitude, or super tropical, and it's also arid, but also happens to have really high species diversity of lizards in particular. And so we've basically been, since basically the time I was a PhD student, I've had a field program collecting specimens and ecological data from arid Australia. And lately we've started working in the Peruvian Amazon. And the general communities that we study there are two of the most species-rich communities on Earth. So the arid Australian community that I work at has about 75 species of squamate reptiles. And in the Peruvian Amazon where we work, we have about 87 species. So we're basically collecting a lot of baseline foundational data on those communities. Now these are very different sorts of communities on some levels because the Neotropics are basically this is basically taking this species richness map overall and basically looking at where lizard and snake diversity is centered. So this is basically looking at the most diverse places on Earth for snakes, and this is looking at the most diverse places on Earth for lizards. So you can see Australia lights up as the global center of lizard diversity, and the Neotropics light up as the global center for snake diversity. Now, one of the reasons this is that I think we need to actually collect lots of field data, and getting back to my general point about whether we have enough data to actually answer some of the questions we would like to answer. This is basically looking at the amount of the relationship between species diversity on a grid cell basis and the number of georeferenced records that are sitting out there in GBIF that we can actually do something with. So the only thing I want you to take home from the only message I want you to take away from this is that the places that have lots of species, like the Neotropics, are also associated with very low density of sampling. So huge tracts of the Amazon basically have almost no data. And of course, this is like the places that have the most species have the least amount of data. So the ratio of data per species is even lower than it looks on this map right here. And so if we want to answer like two of the most fundamental things that you can know about an organism that lets you do anything in terms of ecological modeling that might get at processes that shape the latitudinal diversity gradient are things like where is the species found? This could mean like regionally, where is it found, or where is it found throughout its range? How patchy is it throughout its range? And how abundant is it, and how much does its abundance vary in space? Now, we have those pieces of information for almost 0 species of neotropical reptiles and amphibians. So, except for a very small number of point samples from communities where we have some biased estimates of what those things might look like, we really do not know the answers to these for almost any species of neotropical reptiles and amphibians, let alone all these other things like in some cases we don't know most cases we don't really know what things eat, we don't know how they interact with parasites and predators, we don't know what habitats they use, we don't know how they choose mates, we know almost nothing about their behavior and social biology and so on. So there's a huge knowledge gap here, and I think it really limits what we can say about the causes of the latitudinal diversity gradient. So we've been working in the Amazon, fabulous communities with outstanding diversities of snakes, for example. We've been working in Australia to sort of get at the same thing. You know, if you were to go to a patch of spinifex desert from West Central Australia where we work, you can find up to 60 or 70 species of lizards living in sympatry in the same habitat, which is about up to 10 times more species than you would find in a comparable latitudinally precipitation temperature corrected desert system from North America. So it's fabulously diverse from a lizard perspective. And this is the example of the kind of habitat that you basically find this outstanding diversity of lizards, spinifex sandplain from Western Australia. So this is basically, this is ground zero for the most diverse lizard community on Earth. This is where you could actually find about 70 species of lizards and snakes in sympatry. These are some representative individuals from different clades of squamates from arid Australia. So we go to Australia, we run expeditions out there, we do a lot of pitfall trapping and work with Aboriginal communities as well. And we run large field expeditions to the Peruvian Amazon. It's a lot of hard, muddy field work, involves a lot of trapping with pitfall traps, funnel traps, other sorts of things. A lot of what we do is just active searching at night with headlamps, because a lot of reptiles and amphibians are actually, one of the reasons that we have so few records of some species are actually that they're really hard to sample. In some cases, they're very cryptic. If you look at this image of some Amazonian leaf litter right here, you keep staring at this for a while, you might not actually spot the hemifractus horned tree frog kind of sitting there in the center of the photo. This is a very typical sort of scenario where you spend a lot of time looking at things and not really seeing anything. And so we go and we collect lots of specimens, and one of the things about, all these specimens end up at the Museum of Zoology at the University of Michigan. A lot of them end up split with partner institutions in places like the National Museum of Peru and Lima, for example. And from each of these things, we basically collect a whole range of data that gets accessioned with the specimens. So we have genetic resources, we have lots of different types of genetic material, and we're basically archiving for use by researchers in the future. We've got information for studying disease dynamics. We've got skin swabs and secretions and fecal samples. And so we already have people using these to detect emerging fungal pathogens affecting frog communities, for example, people using our data in publications for that. It's not my primary area of research, but I know a lot of other people want the data. We take snake venom gland samples and skin toxins from amphibians, and we take a lot of quantitative habitat and activity and abundance data as well. And all this stuff basically gets accessioned with our specimens in our collections at Michigan. And I can't emphasize enough that getting these data, and frankly, not just reptiles and amphibian data, but pretty much any group of organisms you can think of in a part of the world that's not super well known or characterized, but getting these data requires lots of hard and expensive field work. And you might look at something like a typical this is hardly a typical Amazonian snake, by the way. It's an emerald tree boa. This is actually a fairly uncommon, spectacular snake. But you could look at a snake like this and say, what does it actually cost to get a snake in the Amazon? Turns out there's actually a published number for how much it costs to catch an average snake in the Amazon, not by us, by another group in 2014, where they did this exact sort of exercise. And they calculated that it costs about $120 to catch a single snake in the Amazon. And that actually works out to about, almost it's almost a spot-on estimate to what we spend to catch a single snake in the Amazon. That's accounting for all the cost of your field expedition and hiring people to help you with, the logistics of getting stuff to remote areas and everything else. But so this is quite expensive. And you think about, now you have a community with like 70 or 80 species of snakes. How many snakes do you have to sample from even one place to reconstruct a species abundance distribution? Thousands of individuals to actually know what a species abundance distribution looks like, and that's just one place, and it's going to cost $120 per snake. So, very expensive sort of, this very daunting sort of challenge that we face. And so I guess my final point is that I think that understanding this tropical diversity puzzle basically requires that we have a renewed commitment to basically collecting and curating natural history data, because it's really the foundation of everything that we do and that we might want to do in the future. And it's even more important in places like the Amazon, where we're basically burning it down around the edges faster than we can actually collect the data that we might want in the future. And so, anyway, that's all I have. So if I have any time, I'm happy to have a discussion or take questions. So thanks for your attention.
Okay, I think we can continue with the discussion now, in contrast to normal Thursday seminars. I have the first question. Why are the speciation rates in fishes higher in high latitude? Kind of obvious.
That's a great question.
Nobody else wanted to ask that question.
So I would say that we don't. I would say that we basically have zero data on that in terms of a causal mechanism. And I like when we were writing that paper, I had co-authors who were like, speculate hard about it, and I pushed back against it because I was like, we have no data on this. Like, however, my speculation would be that, I tend to think about, and you think about like what drives fast rates of speciation? Well, ecological voids, ecological vacuums, ecological opportunity is one very sort of common signal that drives radiations in lakes, on islands, and so on. When you think about this history of disturbance, for example, especially at northern latitudes, and there's been more disturbance than it looks like in southern latitudes as well, in terms of like the Arctic, for example, repeated glaciations, basically scraping off huge amounts of the continental shelf there, massively messed up patterns of oceanic basin circulation happening at high latitudes. So you can imagine that one possibility is that the cycle of disturbance there has driven both high extinction rates, which is consistent with old ideas going back, to Wallace and thinking about what's actually the climatic instability of the high latitudes. But you can imagine that when climatic conditions stabilize to some degree, you also have sort of an engine of speciation in the same way that we see now, incipient speciation, whether it's sticklebacks or other taxa in post-glacial lakes in places like North America and Eurasia, where glacial ice sheets basically scraped everything off. You have new lake systems ready to be colonized by lineages that often form various sorts of divergences into different trophic morphs and so on that are possibly on their way to becoming full species. And so I can imagine that a dynamic similar to that is going on. It's, the data we need to test that would be something that would get at what extinction rates have been. And I'm not sure where we actually get that, but that would be one possible explanation for that. So there'll probably be others. So
wouldn't this be partly recovered by making the difference between the diversification estimates you have from BAM, which are the estimates at present, from what I understood, right? And the diversification estimates you have, for example, from FISA, which are going back through time.
The diversification from what?
The speciation rates from FISA or from the SSE models that you are doing as well.
The rates from
the state of the speciation extinction models. FISA.
The what? No, sorry, I'm not.
FISSE.
Oh, FISSE. Ah, yes. Right. So. You mean thinking about it further back in time?
Yeah, because if it's due to instability, one can imagine that the speciation rates.
You mean rather than focusing on that? You mean
could be slower, but the actual ones could be actually fast if it was really due to the instability in the higher latitudes, right?
Right. So this is looking at it further back in the tree. Yeah. The only issue is that the only thing is that looking at it further back in the tree, we have an issue of what we can actually, so my confidence in those estimates going back in time basically is very low. And so actually I think that, maybe I can't remember if I actually have at one point I had, okay. Right, so this has to do with what I think the information content of a phylogeny is. And so I made this figure in your office today actually. So if you're standing here in the present day looking back in the past, this is sort of what I think of as like the information in a phylogeny in terms of speciation and extinction rates. So if you imagine this is the true amount of information that exists right here, where there's some theoretical amount of information that in principle you could imagine we could extract if we had a perfect record of everything that lived and went extinct. Now, if you look at what we might see looking from today backwards for speciation, our best inferences are going to be closer to the present. It's going to decay as we move back in time. And the reason it's going to decay is because the further we go back in time, the more likely it is that the processes we see today are actually not consistent with the processes in the past because we're just further away in time. So we have less ability to see whether the assumptions that we make when we estimate rates are actually holding. The other thing is that extinction causes all kinds of weirdly prunes out lineages and patterns that happen deep in the past and removes our ability to see things as well. And so it's always had this decreasing function when we look back in time. Of course, even the present day is not perfect also because we have issues like inconsistencies in species concepts and things like that mean that even now we still have a source of bias in those estimates. Now, so that's why I think speciation rates looking back in time are That's why I've started moving away from and trying to over-interpret patterns deep in phylogenetic trees, because I think that I've just been convinced that this is a real issue, and I've generally just been working with sort of recent speciation rates. And I think extinction is something that's a little bit more like this, where it's even quite a bit worse. But it's sort of just a general way I think of thinking about why. Yeah, I mean, we did in our analyses, we did some reconstructions of speciation rates through time, but I guess there's nothing that I take away from that really suggests to me what the causes of that sort of pattern are. So, I mean, we did some we looked at rates through time for tropical and temperate and high latitude lineages. Yeah, it looks like in high latitude lineages you have a fairly recent onset of high speciation. Another problem, though, is when you go deeper back in the tree, this is a really obvious, really serious problem in this particular case, would be that those clades ultimately go back 15 or so million years, and then they basically, through an uncertain biogeographic history, connect to lineages that were who knows where. So at some point, you ultimately you lose your ability to even say this is a high latitude clade, because at some point, those clades that you are saying, hey, this is a high latitude clade, you have decreasing evidence that they are high latitude once you get back beyond like 15 million years or so. And so, where does it actually start being high latitude? So all these sorts of things factor into that loss of information right there. Maybe a tangent from your question, but.
Isn't there any potential systematic bias in the distribution of genetic data? You said you use taxonomy for most of the species, like to several of them, but the genetic data some kind of not other species.
And are you thinking like biases in terms of where we have sampled, like taxa that end up in the data set as genetic lineages? There's definitely biases. There's no doubt that there's biases. So we did a number of things to try to control for that. So one of the things that we did was because we do have some estimates of how many species are unknown or not unknown. We have some estimates, for example, of we know how like the things we use have analytical corrections to sort of account for the fact that some clades are better sampled than others. That said, there's still other ways in which you might imagine those kinds of sampling biases actually playing into our results. So we did a bunch of analyses to look at whether there was consistent effects of sampling, for example, in terms of clades that were well sampled versus poorly sampled. Like Sebasti's rockfish is one of the fast rate clades. That's a well sampled clade where most species have been sampled. Gobies, for example, we can look at a clade like gobies where maybe only 10% or 15% are sampled, and we can look at that and say, well, is there an actual effect of low sampling? And we look at across all the clades, we can sort of account for it. Not account for it, but we can test whether there's a biasing effect. And we didn't see anything. And even when we ask, like, are the undersampled clades that we have, like gobies, even more undersampled? So, for example, our gobies, even though we think we have 15% of the species, what if there's actually, because that's a clade where we actually have even more uncertainty in what the true diversity is, because a lot of them are sort of deep reef, kind of mid, depth kind of taxa, where we have sort of in some ways the least explored elements of the fauna overall. So we did some things to ask whether there were some potential biases there. So we haven't seen anything yet. It doesn't mean there's nothing there, but there's nothing that we've been able to extract from the data that says that there's a signal of bias in that sense. And the clades really are a mix overall of seeing slow rates, for example, in the tropical clade. That's a mix of clades. That's a mix of clades like gobies that are frankly people have just kind of written off, and because they're hard to sample or they're taxonomically difficult. And clades that are like tropical reef, like aquarium enthusiast clades, you might think of like butterfly fishes and things that are much better known because or wrasses where people have been like, oh yeah, like that's a interesting iconic tropical reef clade. So those clades have been much better studied. So, but there's no effect at that scale either.
One more question. I assume that in the north and south, the generation time is much longer than in the tropics. So does it mean that per generation time they have been much longer speciation rate?
And in terms of generation time, I mean, well, that's sort of like the idea of that the theoretical prediction about, I don't buy this, by the way, but the theoretical prediction from like metabolic theory of ecology would be that faster generation times is sort of one outcome one of the outcomes of the effects of temperature on other processes that's contributing to this fast rate of speciation. Now, doesn't seem to be going on. That said, there is an important bias that we did have to look at, which does relate directly to that, which is that generation time actually causes, there's a correlation between temperature and the rate of molecular evolution. This is a big one because you might imagine that could be manifest through generation time. So here's an example of why this would matter. So if in the high latitudes, like the Antarctic and the Arctic, let's say we have both a combination of slower mutation rates and shorter generation times. Both of those things would predict that we would accumulate less genetic change on a given branch for an amount of time. So that would mean that our branches are actually going to be disproportionately shortened at those latitudes relative to the tropics. So this is a very obvious bias that would actually potentially create this illusion of faster speciation at high latitudes without actually being connected to fast speciation. So we had to do a bunch of analyses to sort of try to tease that apart while correcting for a number of factors to just say, well, basically we reconstructed rates of molecular evolution, looked for latitudinal signal and rates of molecular evolution, for example. We don't find any effect there. It doesn't mean that there's not some other weird cryptic way of that's in there, but we haven't found anything yet. But it was an important thing to look for. And that's one of those cases where the bias does go in exactly the way that would look like faster speciation. So.
Is probably very difficult to answer.
Start with a difficult one, and then ask me a second difficult one, so I can't remember the first one.
The point is that we were partly talking about it this morning. That by the term speciation, different people mean different things, right? So if you speak about speciation, it's really something which, as you pointed out, concerns only the tip of the phylogenetic tree. So it is more, it's not like the historical speciation of the lineage, but more something like the contemporary capacity to speciate, given by most recent history, right?
Can I answer that one now? Yes. I would say there's one thing about that though. So any estimate at the tip is basically think about it as a historical rate, but where the weight of the contribution of the phylogeny looking back is a decreasing function of sort of saying. So it's like, if you think about this as a phylogeny rate here, this is almost like the contribution of the phylogeny to that tip is going to be maximally towards like short branch lengths towards the present will contribute more than short branch lengths deeper in the tree. So it is a historical rate. It's just more like, it's just kind of think about it as like averaged over 10 million. Yeah, exactly. It's using that information. It's probably about averaged over about 10 million years.
That was my understanding of it. So the question is that, are you sure it is the same speciation rate as the speciation rate assumed by the proponents of the metabolic theory? If I was a devil's advocate, I would say, okay, but we metabolic theory people mean by speciation something else.
I think it is. So in the sense that, I would say that this is as close to, despite the fact that I just said there is a historical signal there because it's averaged over things, this is nonetheless in some ways the best you're going to do at getting things as close to the present as possible. And if you think about, trying to reconstruct things that are like 30 million years deep in the tree, then that's something where you could imagine it's very decoupled from that. At least in this case, we're trying to get the closest thing that we could have close to the present. The only way you can do that is by using some amount of, if you just use, for example, only the terminal branch, that's if you think about it, the only information that you have that is that waiting time to the last speciation event. But that's so noisy that it's almost like no information in that unless you actually combine it with some kind of weighting information for other sort of adjacent other lineages, basically. So I don't know. I would argue that to the extent that we think, for example, that we're definitely. I guess I would say another way of saying this is that do I think that the speciation rates are pertaining to the time scale over which metabolic theory of ecology speciation rates would be in operation? And I think in some cases, absolutely. So I think that. While the Arctic is a relatively young biome, and some of those lineages haven't necessarily been there for a long period of time, the Antarctic lineages, on the other hand, almost certainly have been there for 20 million years or more. And conversely, many of the warm water tropical lineages have also been there for some time as well. So both of those cases would be where I would expect that to sort of show up, because you would be averaging it then over that even though we have to average it to some degree, you're then looking at it over a scale where I would expect this, where we know we've had warm and colder temperatures. Less clear for the Arctic, but the Antarctic tropical contrast is probably a good one.
Yeah, it's pretty common. And last thing, do you think that this difference between marine speciation rate trends and continental ones are due to the fact that in the sea the productivity is actually much more regularly distributed, or even it is higher in high latitude, so there is like more open space which is like ready to be filled by the speciation?
It's complicated. I have a hard time doing anything with productivity, because productivity in the marine realm is, like if you measure it based on phytoplankton, for example, it ends up showing these peaks that are associated with upwelling zones, which might be at high latitudes. Conversely, though, if you measure it in terms of biomass accumulation, it's going to light up the coral reefs, or you're basically generating lots of these giant reef communities. And so, I have a collaborating on something relating to this with an ecosystem ecologist who does marine fish stuff at Michigan, and so we kind of talk about what does productivity even mean, and it's not so clear, and there's definitely a tendency to just say, well, satellite-based measurements give us this perspective, but that is overlooking what's going on at that sort of scale. But yeah, I guess in the marine system, one thing I would say is that I think there's a key ingredient that is missing from a productivity alone system, alone based argument for things, which is that productivity alone does not help you unless you have structure on which to put it. And I think that structure is bottom. So when you just have high productivity that just kind of rains down, you have nothing to you have no structural features of the system.
Yes, but on the other hand, you have high abundances of fishes in high latitude. So it means that there is this potential, it's like this potential to have many individuals. So basically this ecological space is there. Of course, it's few species, but.
That's true, although there's a lot of other issues there that have to do with where that community actually originates, even in terms of a demographic sense, right? Where are the nursery grounds for that community, and is it an exported community that's basically driven by...
Sure, but I still think that the difference can be that in the terrestrial realm, we do not have peaks of productivity, which is in high latitude, at least, right? So I'm not saying that...
I completely agree. The only thing I would say is that I just don't know. I haven't worked out one way or another how I expect that kind of productivity, including to actually factor into things. But you had a question, right?
Yeah, when you talk about the plant example that you showed, it was consistent with the fish data, right?
Yep.
I didn't get...
For the plant data.
Yeah.
Yep.
That has been published already, or?
That's on bioRxiv, although there was a paper by Alex Antonelli and some co-authors in I actually have this as a...
My question was, I can imagine that the tropical and the temperate
Anyway, it doesn't matter.
would be not equal in terms of proportion of woody
It's
and
fine.
herbaceous.
So what's that?
So that the proportion of the woody and herbaceous species would be different in the tropics.
Oh yeah, no, that's definitely true. Absolutely.
That would be, the generation would be completely different from what Lukáš was asking about the animals, right? So that would have changed the...
So that would be, yeah. So I mean, and to be honest, I don't know if they tested for because there would be life history effects there that so you're getting at whether it would have affected rates of molecular evolution and perhaps secondarily affected reconstructed evolutionary rates. Is that where you were? Now that you mention it, because there is evidence that rates of molecular evolution vary systematically with life history in plants. So I'd be honest that I am thinking about this and the consequences of this for the first time right now, so I don't have a well-defined, and I don't actually know whether they parsed it, but that would be an obvious thing to go back and actually ask those authors to do if their paper has not been accepted yet. So that's a good idea.
Yeah, I have actually a bit similar question to David. So do you think you would see opposite pattern in the freshwater fishes?
In terms of what the pattern looks like for freshwater fishes?
Of the speciation rate with the...
Does not look like that for freshwater fishes.
But is it opposite or is there...
It is opposite, but it's opposite only for one reason. Cichlids. It's cichlids, which I think is so statistically it's not significant, but if you just plot rates, you would see the fact that such a huge fraction of freshwater fish diversity is actually cichlids in basically 3 lakes, really actually 2 lakes, and it's just a massive effect, and their rates are so fast relative to everything else, that cichlids just stand out like crazy. So if you took out cichlids, there would be no effect at all. So if you looked at like New World fishes and you had like, you know, all the big lineages of like piranhas and catfishes and everything, then you basically see no effect. So but the cichlids are this giant, but I'd say that you actually have like, in general, no effect at that scale. So, and you have relatively medium to, you have diverse temperate clades that like persids that just don't seem to be doing anything. And you have some high latitude clades that are still speciating kind of fast, like salmonids, and white fishes, but there doesn't seem to be, it's not anywhere near as strong as it is in the marine realm. So I think there's a lot more to do with it, but it's definitely noisier.
I had wondered, think of it this, so how were these incipient groups that just doing all the hemorrhagic northern boreal or northern hemisphere lakes, are they considered like their speciation rate still pretty low because it's a stickleback, or high because they're just diversifying in every lake they colonize?
So you're thinking about things in northern temperate lakes? In terms of how we consider their speciation rates? So you're asking me like my opinion about what I think about.
Like you did look at that.
Yeah. No, so well.
How these things were considered, because they are too young to really kind of.
No, I mean this is actually. So there was another. Well, I'm tempted to actually get out a slide here, but in the interest of time, I won't do it. But basically, if you take all of those. If you imagine like stickleback, for example, iconic, super rapid speciator, I think this is actually this gets at the heart of what I think is like the central issue with speciation today. And you take sticklebacks and you take everything that is an incipient stickleback form where there's some reproductive isolation or phenotypic differences between populations, and you put those on the overall phylogeny of the Gastrosteidae, the overall family, you will see this phylogeny that basically has lots of, just a handful of speciation events unfolding over 25 million years, and then this massive splatter of all this stuff happening in the last 13,000 years. And what that basically says to me is that whatever we're studying in terms of speciation at the tips of the tree is not really going to be speciation. So almost none of those things that are diverging in the present day have any persistence. They basically don't, they get scraped off by the next glacial event. And they're not, and so it makes me question fundamentally whether if for example, I've estimated speciation rates for stickleback with and without, make it with and without, including, for example, those sort of incipient lineages. And if you were to extrapolate, the rates you would get from including those lineages would be faster than the fastest rate ever recorded for anything by orders of magnitude. So fast that in the space of like 2 million years, you would have like literally the entire Earth. Even with extinction, the entire Earth to a depth of like 100 miles would be sticklebacks. It's like something like 70 species per million years or more per individual lineage, which is, that's like a conservative assumption. So that's an insane speciation rate. Now, the question is the question is if that's actually what speciation rates are like, then why are there species? So sticklebacks also are sitting at they're one of the slowest speciating clades from a macroevolutionary sense. In terms of actually established species, they've done almost nothing. So they're probably in like the 20th percentile overall of rates across all ray fin fishes, that clade. And so what gives there? That just doesn't even, so what it says to me though is that most of those species that are, almost all of those, the probability that any of the divergences we study in sticklebacks are going to go on in 5 million years to be recognized as good species that actually have that longer term persistence is about zero.
Because in 5 million years we won't have any of those lakes anymore.
Well, yeah, but that gets at the critical issue though. So what if part of becoming an actual successful species does not involve simply diverging ephemerally into a little microevolutionary morph, if you want to think about it like that. But let's say all of these significant species that go on to have a sort of a longevity that spans, tens or hundreds of thousands of years, those are maybe a qualitatively different, maybe that's allopatric isolation in the ocean ancestral marine species itself, for example. So if those are very different kinds of speciation events potentially. They're not what we're studying in speciation biology for the most part, but that could be, so it could be that the species that actually become the species that we would see in that long term are originating via a very different sort of process. And it might be that, you know, if any of those sticklebacks that are forming in these little lakes that get scraped away by a glacier actually manage to expand their geographic ranges back out into the marine realm, then they probably wouldn't go extinct, right? So that suggests that there's whatever's keeping them from expanding their ranges back into the marine realm to be sympatric with their ancestral species. There's probably it's an interesting control there. So it's a challenge. I think the time scale over which we'd be measuring it would probably be a bit too far to see any sort of impact at that scale. So.
And another one, I don't know much about fish. So, is there any correlation regarding species richness and or speciation with size of the fish?
With what?
Size of the fish.
With the size of the fish. That is a good question. So overall, I would say no, but it's definitely like, I mean, if you look, I mean, there's kind of like like large fishes in general aren't. I mean, the clades of fishes that become really large and really small tend to not be these clades. So this is kind of, if you looked at the body size distributions overall, it's kind of in the kind of central actually. It would span a pretty large variety of things. They wouldn't tend to be the larger bodied fishes, but there's not a clear connection to size though. Definitely not a strong connection to size. That's a good question though, and there are theoretical reasons to think about how that might play out too, and it relates to things like generation time and so on. But yeah, but no strong effect there for sure.
No? So thank you again.
Thank you.